Reverse transcription, reverse transcription, reverse transcription, reverse transcription.
Hi, do you want to be the very best, like no virus ever was? Then you're in luck, because today we are going to talk about a technique called reverse transcription, which you should know if you want to conquer the world with, of course, many clones of you. It is a technique used by the glorious, magnificent families like Retroviridae. In that family, there are famous viruses like HIV. It's one of our most successful viruses, truly an idol. Viruses like HIV use reverse transcription. In short, in reverse transcription, cDNA is produced from the viral genome and the double-stranded DNA is produced from the cDNA. Viruses like retroviruses inject its genetic components and enzymes necessary to hijack the host cellular machinery, the most important of which are viral genome RNA and enzymes like reverse transcriptase. In this video, we will be teaching you how this reverse transcriptase works so that you can conquer the world. This reverse transcription takes place in the host cytoplasm, and this is the viral genome RNA. A tRNA will act as a primer and it will bind complementarily to the primer binding site, or PBS for short, in the viral genome, and this is the starting point of the cDNA. The 5' to 3' direction for the viral genome is from this to this, and the 5' to 3' direction for the cDNA is from this to this. After the primer has been bounded, the reverse transcriptase will transcribe the DNA nucleotides from the 3' end of primer complementarily to the RNA genome from U5 until the end of the R region. Moreover, the reverse transcriptase has also RNase H domain, which is an RNase, which means it can degrade RNA molecules. Therefore, after the complementary strand from U5 to R region of cDNA has been completed, RNase H of reverse transcriptase will degrade the U5 and R region of the viral genome. In addition, there are R region on the both sides of the viral genome. tRNA primer will then jump to the other side of the viral genome, and the R region on the cDNA will complementarily bind to the viral R region. Then the reverse transcriptase will transcribe from the R region of the cDNA according to the viral genome. Then the RNase H of reverse transcriptase will degrade most of the viral genome except for this PP sequence. The remaining PP fragment is then used by the enzyme as a primer to initiate the transcription of the second DNA from 5' to 3' direction. The tRNA primer then leaves the cDNA. The jump happens again, and the PBS of the cDNA is complementarily bounded with PBS of the second DNA. The reverse transcriptase synthesizes DNA nucleotides from the 5' to 3' region of both cDNA and the second DNA. So this is the process how you can create from viral RNA to complete functional double-stranded DNA. Now you have seen how our reverse transcription works. Don't you think we are really awesome? Our viral evolution is amazing. We are technically not alive, yet here we are, becoming really successful. Especially our latest sensational virus, the coronavirus. Long live the pandemic! Earth-chan, eat that corona-sama! But there's one thing that really pisses me off. Our evolution is exploited by these humans. Well, to use against us, let me show you how. These humans created something called a real-time reverse transcription polymerase chain reaction, or real-time RT-PCR for short. In real-time RT-PCR, they extract our genome from us, which results in rupture of our membranes, killing millions of us, these barbarians. Ugh! After they obtain the viral genome, they put it into a test tube, which contains primers, reverse transcriptases, tag DNA polymerases, deoxyribonucleotides, and fluorescent probes. At the first step of the RT-PCR, reverse transcription of the viral genome is done by the reverse transcriptase, which creates a complementary DNA or cDNA of the viral genome from the added free nucleotides. The initial denaturation is carried out by increasing the temperature to about 95 degrees Celsius and causing the denaturation of the RNA-DNA hybrid. The initial denaturation destroys the reverse transcriptase enzyme with the heat, but Taq DNA polymerase can withstand the high temperature because the enzyme is extracted from a bacterium that is evolved to survive in hot springs and hydrothermal vents. So their proteins like enzymes can withstand such high temperature, but virus enzymes cannot withstand. After the RNA, cDNA template has been denatured. In the annealing step, the temperature is lowered to 58 degrees Celsius, which allows the annealing by complementary binding of primers to the cDNA and Taq DNA polymerase transcribe DNA nucleotides from the 5' to 3' direction by adding free deoxyribonucleotides, creating a double-stranded DNA. In the denaturation step, the temperature is then increased again, which creates two single-stranded DNAs and primers will bind to them. The fluorescent probes are strands of nucleotide that will bind to the DNA strand that has the same orientation as the viral RNA. The binding will result in emission of signal, which is responsible for the detection of the presence of the viral genome. PCR is very sensitive because if there is only one strand initially and after 30 cycles of PCR, in the end there will be about 1 billion strands of DNA. Therefore, these humans can detect us from very few strands of our genome. Do you know what I'm talking about? This is what I'm really angry about. This is exactly like this. What's happening?
Dame, dame, dame